Why would these researchers want to shock trout with backpack tasers, implant them with little radio stations, and then make them toss their cookies in a pan? It's all part of a large experiment at Hinkle Creek in the Cascade foothills, just east of Sutherland, Oregon, where scientists are inquiring into a complex puzzle that could affect the way we manage Oregon's forests. Oregon's population is growing, and our demand for wood products is growing with it, putting more pressure on our foresters to provide the wood we need while still protecting environmental values like clean, abundant water and wildlife habitat. Oregon has been a pioneer in the use of science to help manage our forests. In 1971, Oregon led the nation when lawmakers created the Oregon Forest Practices Act. It used the best science of the day to regulate logging and forest road building to protect our environment. But harvest practices have changed dramatically in the 30 years since the act was created. New technology is much lighter on the land. Road construction methods have improved, and land managers are even more environmentally aware. Meanwhile, advances in science and technology have given us a far more detailed understanding of these complex systems we call forests. To update the Forest Practices Act for the 21st century, scientists and land managers have teamed up to conduct a 10-year study at Hinkle Creek to measure just how modern forest management activities affect our watersheds. To do this, the scientists at Hinkle Creek first needed to design their experiment. Hinkle Creek is a near-perfect setting for a big forest science experiment called a paired watershed study. A watershed is an area of land that's drained by a river or creek system. Hinkle Creek has two neighboring watersheds, the North Fork and the South Fork, that are very similar to one another, and together they cover 5,000 acres of prime forest land. The landowner, Roseburg Forest Products, saw an opportunity to help scientists study the impacts of modern forest practices. They agreed to leave the North Fork untouched for 10 years, the control, while conducting harvest activities on the South Fork, the experiment. Scientists will compare the two watersheds to evaluate the effects of logging versus no logging on a spatial and time scale never before available. But where do you begin such an ambitious task? The scientists can't just go out and start measuring things. They first need to figure out what questions to ask, and to ask them in a way that could be answered through scientific inquiry. Historically, fish have been a reliable indicator of the health of a forest watershed system. Logging, road building, and forest management activities can affect the fish in a number of ways, both good and bad. Trees use a lot of water. Removing trees for harvest could put more water into streams, which could increase the drift of insects downstream and be good for fish. Or it could cause erosion and put more sediment in the stream, which can damage fish habitat. So the Hinkle scientists settled on the question, how are fish affected by logging operations on the watershed? To observe meaningful changes that occur to the fish population, the researchers need to identify the variables. But which variables? Since logging might have both positive and negative impacts on fish, an obvious variable would be abundance. That is, how many fish are there in Hinkle Creek before and after logging? But is abundance a meaningful variable?
Studying fish is sort of a complicated process because you're dealing with a mobile organism. Sometimes abundance can be sort of a tricky variable because people oftentimes assume that because there are more fish somewhere that equates to better habitat. And there's all kinds of things that go on with fish just like they go on with people. Like if you were to look at a city and you didn't know anything about people and you were trying to figure out where the best habitat was and you randomly selected some sites to study, One of your random sites might be a dorm. There would be a ton of people in a dorm, but are dorms good habitat? So it becomes a little bit tricky, and the same kinds of things can be going on with fish. Fish can be territorial. You could have the biggest cutthroat in the creek sitting in the nicest pool. It'd be like the bully on the playground, and it would tend to push other fish out. And you went there and sampled that, and you'd get one fish. And if you were just looking at density, you would assume, ooh, that's not very good. So it's a tricky thing to get at.
Because looking at abundance alone can be misleading, the scientists add new questions. Does location affect abundance? And if fish go where the food is, does diet affect abundance? Now that the questions are defined and the investigation designed, the team collects the data. The questions the scientists chose requires them to track, count, and measure the fish as they move around the watershed. To know the location, the experimental design had to include a method to follow the trail of individual trout. A small electronic device called a PIT tag is the answer. PIT stands for Passive Integrated Transponder. It's a tiny microchip transmitter that's activated when it passes near a scanning device. This gives each fish a unique electronic barcode that can be scanned as they move around the watershed. Now, all they need to do is get the PIT tags into the fish. First, technicians do some electrofishing. These Ghostbuster backpacks generate electricity which temporarily stuns the fish, making them easy to net.
This one doesn't have a pit tag because he's not clipped, so he will get a pit tag because he's over 100 millimeters.
Next comes minor fish surgery. In a quick and painless procedure, the small pit tags are inserted into the abdomen of each fish.
It's a really small incision. It's about 2 millimeters long, and it looks pretty rough, but it doesn't seem to hurt them at all. They're a lot tougher than we are.
This is also a chance to collect data on size, so the fish are measured, weighed,
192.
and released back into the stream. Meanwhile, electronic gate readers are installed at numerous locations in the watershed. These radio antennas are stretched across the stream and are hooked to sensitive electronic counters. When a tagged fish swims through a gate reader, a mild electronic field around the antenna causes a fish's pit tag to broadcast its information to the data recorder, much like scanning items at a supermarket checkout.
When those fish swim through one of our antennas, we know that that's Joe or Jill or whoever. And we have these antennas set up all over the watershed, one at the downstream end of each watershed and then one at the mouth of each fish-bearing tributary, so that we can tell who goes in between these areas of the stream.
Technicians also use mobile antennas to track fish after they've moved through the gate readers to know precisely where they're hanging out. Knowing where the fish are going gives valuable information about potential effects of logging activities.
It allows us to see, without really harassing the fish, whether they are selecting different kinds of habitat. Like if you have an acute response, like say logging happened and it changed something in one of those tributaries and all those fish said, whoa, this place is not good anymore, and they left. Or if it made it really good and all the fish down in the main stem said, geez, there's a whole bunch of bugs coming out of that trib, and they all move in there. Well, the gate reader is what would tell us that happened, and it would tell us exactly when it happened.
To investigate the relationship between location and diet, the scientists also need to know what the fish were eating. This time, the technicians revisit the streams, recapture the tagged fish, and collect samples of what the fish have in their stomachs, in a procedure politely called fish gacking. Basically making the fish throw up their last meal for analysis in the laboratory.
Looks like half of a millipede. Cool.
So let's review the process. The initial question of how are fish affected by logging operations generated a list of additional questions to investigate, variables to test, and data to collect. The next step is displaying and analyzing the data. When you have to analyze so much information, a graph can be worth 1,000 data points. Graphs can display data in a way that helps the scientist recognize patterns and conduct their analysis.
We use some geographical information system tools, particularly some modules in a program called ArcMap. It moves up the stream and it says, well, how many fish are there? And it sort of calculates a relative estimate of that. We can color code it so that the hotter colors are associated with more fish and the colder colors are associated with fewer fish. And so we can kind of make a density map of the whole watershed. You can just look at those maps and you can see exactly where fish are hanging out and where they're not hanging out. And so the idea is to sort of create this map of distribution and look at it for the years prior to timber harvest, and then start building a map looking at distribution after timber harvest. It's like your eye is this incredible integrator of information. To communicate that information in a numerical format is very difficult, but when you see the maps, you just go, oh, there's the spot. And it's a pretty fun thing to do and pretty intuitive. It's not very difficult to see the response of the fish.
When they looked at those responses, patterns emerged, and so did some surprising discoveries about the lives of fish.
We can tell right now that we have fish that never go anywhere. Basically, that they emerge from the gravel and they find a little rock and they live under that rock pretty much for their whole life. We have other fish that leave and we never see them again. They go all the way down to the Umpqua, maybe to the ocean. It appears that they're a lot like people in a way, in that they have personality types. There's people who are risk takers. They'll go someplace they've never been at the drop of a hat. There's people who are very conservative that, you know, it takes a stick of dynamite to get them to go anywhere. And these same sorts of traits are apparent in these fish.
In interpreting these results, the researchers begin to understand that the real key to the survival of cutthroat trout lies in how many different kinds of fish personalities there are. These personality types can be measured by analyzing fish behaviors observed in their environment.
We see all of these different behaviors, and as scientists, we think that it's important that these behaviors persist through time.
The trout develop these diverse behaviors over time, enduring things like ice ages, droughts, and extreme wet periods.
You know, over the course of their evolution, they've seen a lot of stuff.
And having survived a lot of different kinds of environmental stuff has kept the population diverse and resilient to other changes.
The diversity of behaviors in these organisms sort of buffer them from impacts and how they might respond, like to some disturbance like logging, and how they persist through long periods of time.
So the question of how logging affects fish became an inquiry into abundance, size, and distribution of fish in Hinkle Creek. But that in turn evolved into a much broader and perhaps more interesting question about the trout's ability to persist through time. But that's the nature of good scientific inquiry. It's an organic process. As you find out more information, the problem changes, and that leads you to new questions.
It's a real iterative process, and the one thing that's interesting about research is that everything you do is pretty much something that you've done for the first time in your life. So you're always figuring things out, and you usually never figure it out just right the first time. So everybody is constantly reevaluating and adjusting what they do. Science is a way to kind of learn in an organized manner, I think. A lot of people think that you prove things, but you basically never prove anything. All you do is show that something is not it, and then you move on to the other possibilities. And over time, you know, the weight of evidence just starts leading you down different tracks.
The inquiry at Hinkle Creek is far from complete. There are many more questions to ask, variables to measure, and data to gather, analyze, and interpret. The researchers are confident their findings will eventually tell us if there are any changes we need to make to better manage our forests. At the same time, they hope to also give us a grander understanding of those who live there.
People, I think, assume that we know everything about fish, and I'm here to tell you that we don't know much. Every year we see a behavior by a fish that makes all of us just go, "Huh, I didn't know they could do that." I mean, it's crazy. But it's part of what makes it really fun to do this, is that you're just constantly totally surprised.